Peptide-Oligonucleotide Conjugates explain the next big thing in targeted drug delivery. You have spent months, maybe years, optimizing your oligonucleotide therapeutic. The sequence is perfect, the biology is solid, and then it hits a wall. Not because the molecule doesn't work, but because it can't get where it needs to go. That's not a rare scenario, that's the defining challenge in RNA therapeutics right now. And today we are going to talk about technology that's challenging that equation. Hello, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories. We work with pharma and biotech companies at the analytical and development for frontier of the complex molecules, including conjugate-based therapeutics. Why traditional oligonucleotides struggle? Here is the fundamental problem. Oligonucleotides, your siRNAs, antisense oligos, splice switching candidates are large negatively charged molecules. The human body, quite frankly, was not designed to welcome them. They get degraded rapidly in serum, they get cleared by the kidneys before reaching their target, and even when they do arrive at the right tissue, getting into the cell is an entirely different pattern. GalNAc conjugation solved part of this for the liver, but what about the brain, the muscle, tumor cells? That's where the field has been stuck. And that's exactly where peptide oligonucleotide conjugates comes in. So what are peptide oligonucleotide conjugates are? So what exactly are we talking about? A peptide oligonucleotide conjugate, or POC, is precisely what it sounds like, a therapeutic oligonucleotide that has been covalently linked to a peptide. That peptide isn't a decorative, it's the functional. It acts as a delivery vehicle, a targeting agent, sometimes a cell-penetrating guide, steering the oligo to where it needs to be and helping it get inside the cell. Think of it like this. Your oligo is the drug, your peptide is the GPS and key combined into one. How POCs improve drug delivery? These architectures addresses the three biggest delivery bottlenecks simultaneously. Number one, tissue targeting. Cell-penetrating peptides or receptor targeting sequences dramatically improve tissue specificity, particularly for muscle, CNS, and oncology targets where other delivery vehicles fall short. Second, cellular uptake. Peptides actively facilitate endosomal escape and intracellular trafficking, a perennial headache in RNA delivery. And third one is stability. The conjugate often adds a layer of protection against nucleases, extending the molecule's half-life in circulation. The result? Better efficacy at lower doses, and for patients that matters enormously. Chemistry behind peptide oligonucleotide conjugates. Now this is where it gets technically interesting, and honestly where most development programs underestimate the complexity. Peptide and RNA chemistry are fundamentally different. They require different protecting group strategies, different synthesis conditions, different protection steps. When you're trying to bring them together in a conjugate, you have to sequence every reaction precisely to avoid damaging either component. Protecting groups are not optional, they are critical. One wrong deprotection step and you have compromised your yield, your purity, or both. Then what's the linker chemistry itself? Bioorthogonal coupling strategies that are chemically selective and biocompatible has been a game changer here, allowing chemists to attach peptide to oligos without triggering off-target reactions. At Resolve Mass, one of the things we have invested heavily in is the analytical infrastructure to characterize these complex conjugates. High resolution mass spectrometry, robust method development, because if you can't confirm the exact conjugate structure, you don't really know what you have made, and regulators will ask that. Why linkers matter? Let me spend a moment on linkers specifically, because this is often underappreciated. The linker isn't just a bridge, it determines the molecule's biological fate. Is it cleavable, under what conditions, intracellular pH, enzymatic activity in the target tissue, does it release the oligo in its active form? A poorly chosen linker can neutralize an otherwise brilliant design. A well-designed linker becomes a controllable release mechanism. This is where the chemistry meets the biology, and where experienced teams make decisions that inexperienced ones don't even know where they need to make. Where peptide-oligonucleotide conjugates are being used. Clinically and in late-stage development, POCs are showing up in some of the most high-value, high unmet need areas in the medicine right now. Neuromuscular disorders, where getting therapeutic oligos past the blood-brain barrier or into muscle tissues has always been the bottleneck. CNS applications, precision oncology, rare genetic diseases, all areas where current delivery platforms have real limitations, and POCs are starting out to fill the gap. If your pipeline touches any of these indications, this platform deserves serious consideration. Let's talk about manufacturing and quality challenges. Here is the part that doesn't get enough airtime in the excitement around POC biology. Scale up is hard. These are not simple molecules to manufacture. Yield consistency, purity profiles, impurity characterization, all of these are significantly more complex than a standalone oligo. GMP compliant production requires rigorous process controls at every step, and the analytical methods to support it have to be purpose built. This is precisely the space where a specialized CRO can compress your timeline and reduce risk. Not because the science is impossible, it's not, but because the learning curve is steep, and doing it wrong costs far more than doing it right for the first time. For IND and ANDA programs especially, regulatory scrutiny on a complex conjugate is increasing. The data package needs to be airtight. So let's talk about key takeaways of this session. So let me pull all this together. Peptide-oligonucleotide conjugates aren't a distant future technology. They are in active development right now, addressing real limitations in RNA delivery that the field has been wrestling with for a decade. The peptide enables targeting and uptake. The linker chemistry determines the therapeutic outcome. The manufacturing and analytical complexity is real but manageable with the right expertise. And as precision medicine continues to expand, POCs are positioned to become a core delivery modality across multiple therapeutic areas. If you are working on an oligonucleotide program and delivery is your bottleneck, this is the conversation worth having with Resolve Mass Laboratories. If any of these resonated, whether you are an early stage exploring delivery options or deep in development, hiring analytical and manufacturing walls, We genuinely enjoy the conversation. Drop your questions to our team. If there's specific aspects of POC chemistry, linker design, or regulatory strategy you'd like us to go deeper on, let us know. We'll build it to the next one. And if you found this useful, share it with the colleague who is working through these challenges. This is the community that benefits from better information. Thank you so much for watching this session, and see you in the next one. Until then, goodbye for now.